It is my great pleasure to introduce Dr. Christine Ng. Dr. Ng is a professor of molecular and human genetics and the Chief Quality Officer and Vice President and Chief Medical Officer of Baylor Genetics. Her main research expertise is in lysosomal storage diseases and in whole exome and whole genome sequencing. She's been a leader here at Baylor, and her work has shown that diagnostic genetic testing not only has value today, but leaves a legacy of data that can be used to solve for undiagnosed diseases that are even yet to be discovered or defined. Her work has rapidly translated into benefits for patients and has set new standards for the practice of medicine. Congratulations to Dr. Ng. I invite you to learn more in this video presentation on her recently published work.
Thank you very much. It's an honor to be recognized today by the DeBakey Medical Foundation for the work of myself and my colleagues over the past 3 years. It's especially meaningful to me as I am a graduate of the Tulane University School of Medicine, as Dr. DeBakey was. And we had the honor of having him be our commencement speaker at the medical school graduation. And I can't think of a better role model, educator, surgeon, and physician to send the graduating class off to start their medical careers. So again, thank you very much for the recognition. The research that I'm going to be speaking about today is around the subject of sequencing approaches to the undiagnosed disease patient. And I've had the honor of working with many talented colleagues around the country and locally here at Baylor to produce the following 3 papers that are being recognized today. The first is an NIH collaboration, multi-center, for the Undiagnosed Diseases Network. And this work was published in the New England Journal of Medicine. The second is locally by my group at Baylor Genetics, and it's about reanalysis of exome data to increase the diagnostic rate. And it was first authored by Pengfei Liu. These 2 papers had recognition at the end of 2019 in an article published in the American Journal of Human Genetics and senior author by Eric Green from NHGRI as 2 of the top 10 genomic medicine articles of the year. The third article, first authored by Jinglan Zhang, is a non-invasive approach to screening pregnancies for de novo dominant conditions. And I'd like to tell you a little bit about each of these papers. The genesis of our work in whole exome sequencing for the diagnosis of genetic conditions started in 2011 when we launched exome sequencing in clinical practice. We published 2 articles, and Yaping Yang was instrumental in these articles. The first was our experience with the first 250 cases of exome conducted here at Baylor, and the second was a follow-up on a much larger cohort of over 3,000 cases. We've done over 16,000 total clinical cases of exome sequencing, and this work led to us being selected as the sequencing core for the NIH Undiagnosed Diseases program. As I mentioned, this is a multi-institutional NIH-funded group of investigators that are using both traditional and novel evaluations of patients with suspected genetic diseases. Patients are invited to clinical sites where they're evaluated in a multidisciplinary setting, so specialists in genetics, in neurology, and other subspecialties all work together to try to come up with an approach to diagnose these challenging patients. They're able to utilize all diagnostic modalities that are available in a hospital setting, but then the grant also enables them to use core facilities. As I mentioned, Baylor is a sequencing core for the exome and genome sequencing. There is also a model organisms core led by Dr. Hugo Bellin here at Baylor, and a metabolomics core. And in addition to diagnosing patients with known genetic conditions, the network is also focused on new disease gene research. This is a map of all the different clinical sites. As you can see, they are spread around the country as well as the cores. In terms of our process of offering exome or genome sequencing to the UDN network, the network is able to choose if they think exome or genome is more appropriate for the particular patient. We sequence both the proband and family members at an average of about 3.5 individuals per UDN proband. And then the sequencing core uploads the raw data within 2 weeks. We also provide a clinical interpretation and report that is suitable for inclusion in the medical record, and the clinical site does their own interpretation as well. We also augment the exome or genome sequencing with research RNA-seq to help prioritize variants. In terms of making diagnoses by different modalities, the overall diagnostic rate for this cohort of patients was about 35%. And as you can see, exome or genome sequencing accounted for the largest percentage of diagnoses, so it was able to facilitate about 74% of those diagnoses. As I mentioned previously, one of the objectives of the UDN is also to discover new disease-gene relationships. And you can see from this slide 4 different examples of evaluation of UDN patients leading to new disease-gene discoveries and publications. In terms of where the UDN stands today, we've received over 4,000 applications for patients wishing to be evaluated by the network. About 4,000 have been under review, and of those, about 1,600 have been accepted for evaluation. So far, about 1,392 participants have been evaluated, and about 400 have been diagnosed. So that's a diagnostic rate currently of about 30%. One very interesting aspect of exome and genome sequencing that has recently come to light is reanalysis of existing data. And so we have shown, and Pengfei Liu in the group has taken the lead in this, that reanalysis of existing genomic data can improve the diagnostic rate over time. So as I mentioned, the diagnostic rate with the UDN is currently at 30%, but if you were to reanalyze the data over time, that diagnostic rate could very well increase. And I can show you some examples of this. As I mentioned, there was a paper with our initial 250 patient experience with clinical exome sequencing. And if we took a look at those 250 patients and reviewed the data again using today's knowledge base, or in this case, the knowledge base from December 2017, we were able to increase this diagnostic rate from the initial about 25% to a diagnostic rate in 2018 of about 47%. So a significant increase. 60 new cases received a molecular diagnosis. So this was after the initial sequencing. They are then contacted and told that by reanalysis of their existing exome data, we're able to then provide them with a new diagnosis. Of these cases, 60 new cases, 75% was because a new disease gene was discovered in the interim. 5.9% was because a variant was reclassified using new information. And then there were several other categories, including finding copy number variants that would augment the single nucleotide variants discovered by the exome. Also, a reanalysis of another cohort, and as I mentioned, there was a second cohort of about 3,000 patients published in JAMA, and something very similar was discovered, but this is a shorter time period to the reanalysis of this group. So the diagnostic rate for this group increased from 25% to about 36%. And again, new disease genes accounted for the majority of these new diagnoses. There are several unique properties of exome reanalysis that make it unique in medical practice today. First of all, there is no or very minimal wet bench laboratory work that is incurred for generating NGS data to be reanalyzed. Either the laboratory can perform this reanalysis or the physician can request it directly. It can occur at any time after the initial clinical report is issued and potentially can be repeated at specific intervals. The return of reanalyzed results does require communication between the laboratory and the referring physician to make sure that they're still caring for that patient and can utilize the new results in the best possible way for patient care. So in summary for this part, the use of whole exome sequencing in clinical medicine can help solve the diagnostic odyssey for patients. Clinical genomic sequencing in the rare disease population can facilitate new disease gene discovery. In addition, and as we have shown in other publications, the early application of genomic approaches to the undiagnosed patient can lead to better informed medical care, particularly for NICU and PICU patients and other critically ill patients. And then reanalysis of existing genomic data can improve the diagnostic rate over time. For this part of the presentation, there are many individuals who I'd like to acknowledge, particularly Brendan Lee, Richard Gibbs, Art Baudet, Sharon Plon, and Jim Lepsky, as well as Yaping Yang, who are really instrumental in the development of the exome sequencing protocol. I'd now like to discuss this paper led by Jinglan Zhang and Jianli Li from our group at Baylor Genetics. And it describes a non-invasive prenatal sequencing test for multiple Mendelian monogenic disorders using circulating cell-free fetal DNA. This test was developed to address an unmet need in prenatal screening. As we know, there are multiple approaches to determine a couple's risk of having a pregnancy affected with a genetic condition. One of those is by universal carrier testing, which is widely available for autosomal recessive and X-linked conditions. Secondly, there is non-invasive prenatal screening, which can determine the risk for having a child with trisomy or microdeletion or microduplication. But there is an unmet need for autosomal dominant de novo conditions for which there was no approach for prenatal screening. As we can see from this study that was conducted several years ago, but looking at more than 1 million consecutive live births, the incidence of chromosomal abnormalities was 0.18% of total births, whereas the incidence of dominant conditions was 0.14, which is relatively close to that of chromosomal abnormalities. And we also know from our experience with whole exome sequencing that over 50% of cases that were diagnosed by exome sequencing in the pediatric population were autosomal dominant conditions. And of those, 74% were de novo. So again, there is an unmet need to screen for these potentially devastating conditions in the prenatal setting. The genes that we included in this non-invasive multi-gene panel are shown here. Individually, they're somewhat rare, but if you take a look at them cumulatively, the incidence is about 1 in 600. And they're divided into these different categories of Noonan spectrum disorders, skeletal disorders, syndromic disorders, and the selection criteria for the disorders included in this panel were that they tend to occur de novo. And as we know, with advanced paternal age, genes such as FGFR2 and FGFR3 are often involved. Also, we wanted to select genes with a clinical impact. So there would be a need for prenatal, perinatal care and management, such as with the skeletal dysplasias, perhaps early surgical or medical intervention, perhaps with the Noonan spectrum disorders would be indicated, as well as some disorders with cognitive disability. This is the workflow for the test. It does require samples from both the mother and the father, and we isolate DNA from the maternal plasma for this circulating cell-free DNA, and also genomic DNA from the mother and the father, and it undergoes NGS library preparation as well as next-generation sequencing. And then the data is analyzed for variants of low allele fraction. Looking at the indications for the samples that had been submitted in this particular series of over 1,000 cases, about 48% had advanced maternal or paternal age, and about 31% had abnormal ultrasound findings. And you can see other indications such as family history or a father who was affected with one of the 30 disorders. And you can see here our positive findings. We had 23% diagnosed with thanatophoric dysplasia, very severe skeletal dysplasia, and about 22% with one of the Noonan spectrum disorders, 12% with achondroplasia, and 26% with osteogenesis imperfecta. Since this is designed as a screening test, we did recommend that the results from the non-invasive test be confirmed by invasive testing, and this shows that for cases that did go on for invasive studies, the diagnoses were confirmed. There were several cases in which the family opted not to have an invasive procedure because the phenotype as well as the non-invasive testing were concordant. When a new genetic test is being designed, the indication for testing is considered as well. In this case, the indications for testing were thought to be advanced paternal age, abnormal ultrasound findings, as well as couples who wanted to know as much information about their pregnancy as possible. When the test was launched, we in fact saw some cases submitted of fathers who were affected with one of the genetic conditions on the panel. One of the surprising uses of this test in clinical practice turned out to be couples in which the father was affected with one of the conditions on the panel, and the couple wishing to know what the risk of passing the condition on to a child was. And so these cases that were submitted for the non-invasive screen showed that the father indeed had a variant in one of the 30 genes on the panel, but that the plasma findings in these particular cases were negative, indicating or suggesting that the father did not pass on the condition. We did a clinical outcome study as well, following up a cohort of over 200 cases who underwent this test clinically. And we did not find any discrepancies between the results of the non-invasive test and the pregnancy outcomes. So to summarize, we developed a novel test for non-invasive screening of 30 genes associated with autosomal dominant and X-linked genetic disorders, which tend to occur in a de novo fashion. Our early experience demonstrated a positive rate of about 5.6% in a relatively high-risk cohort with a high number of cases with ultrasound abnormalities. And our early clinical experience with this test demonstrated utility in patients with prenatal ultrasound abnormalities, fathers affected with one of the disorders, or couples desiring additional prenatal options. And I'd like to acknowledge these individuals who contributed to this project, most importantly, Jinglan Zhang, as well as Inyan van den Wever. Thank you very much. And again, thank you to the DeBakey Medical Foundation for this recognition.